All right, thanks for having me today. Happy to talk about an update on CAR T-cell therapy for multiple myeloma. All right, these are my disclosures. Today we'll talk a little bit about how CAR T-cell therapy works, the data for the initial studies that led to approval in the late-line therapy of myeloma, some of the more recent studies that led to approval in the early-line therapy, early relapse therapy, and then just a little bit of discussion about who needs CAR T and what are some of the factors to use in deciding when and who to use it for. Just these are the 20 different therapies that have been approved for multiple myeloma since 2000. But I just wanted to point out that the last 5 have all been T-cell based therapies. So either T-cell redirecting bispecifics or CAR T-cell therapy. Before we go through all these data and trials, I just wanted to go through a case of why this is important. So we have a patient who was 61 at diagnosis, over 73 now, diagnosed back in 2012 with stage 3 myeloma, had double hit disease with 17p deletion, AD1Q, had 9 lines of therapy over the course of about 6 years. With all of the different alphabet soup combinations listed there. Her light chains rose to the 500s, normals less than 19 or 20. And then, there we go. At that time, there were no CAR T-cell therapies available, so she actually had to travel all the way from Minnesota to Texas to get a slot on a clinical trial. So just a few years ago, this was how hard it was to get these therapies. So she had to come all the way to Texas. We collected her T cells in April of 2018. We gave her some bridging therapy with things that she had already failed, things that already failed her, and then kept a lid on the myeloma for a month. It takes about 5 weeks to genetically modify those T cells, and then got the infusion in May of 2018. With ide-cel on the KarMMa trial. She actually had grade 1 cytokine release syndrome, which is pretty typical, although the median onset with ide-cel is day 1. Hers was not until day 14, so it delayed the hospital discharge. So then ended up being in the hospital until day 18. Did not require any antidote like tocilizumab or steroids, went away with Tylenol. No neurotoxicity. And interestingly, her light chains didn't drop until after that fever, which is a signal that those T cells are expanding and acting. And then by 3 months, she was in a stringent complete response. And at 6.5 years, she's still in a complete response and not needing any therapy other than some intravenous immune globulin replacement for hypogammaglobulinemia. Because as Dr. Afro mentioned earlier, the CAR T cells, just like bispecifics, target all of those plasma cells and precursors with BCMA. So what is a CAR? What does that mean? So CAR stands for chimeric antigen receptor T cell. So a chimera was a Greek mythology monster with the head of a lion and the tail of a dragon or something like that. And so in this case, we have the, sorry, can't find there's the pointer. Yeah, so the head of a monoclonal antibody that targets the surface antigen on the top of the myeloma cells, and then the tail is a T cell receptor that causes signaling inside those T cells that forces them to attack and kill the target. And so it's a completely hybrid structure because the normal T cell receptor here is completely much more complex. It actually can't see an antigen protein on the surface of the cell. It requires the cancer cells to chop up their antigen and present it as a little tiny peptide in the groove of major histocompatibility complex, and many cancer cells don't express MHC, and so often T cells can't ever recognize the cancer. And so in this way, we're essentially taking a T cell from the patient and forcing it to recognize their cancer. This is another way of looking at it. So we have a T cell from the patient that we collect, ship off to a company. The ones that are currently in practice use either a retrovirus or lentiviral vector to insert that DNA into the DNA of that patient's T cells. That gets incorporated, so then it gets turned into protein that gets expressed on the surface of the T cells. And then when they're infused into the patient, can go in and attack any cell that has that target throughout the body. This is another way of looking at it. The machine that we use to collect these T cells is the same machine that we use for autologous stem cell collection. It's just different. Inside, they collect out different cells. They collect out the T cells instead of the stem cells. And it takes like a half a day to collect. We don't have to give any growth factors like we do with stem cells. And there's no chance that they have to come back a second day like with stem cells. So it's easier. But then they get shipped off to whichever company's making that particular product. They get genetically modified, expanded typically to hundreds of millions of cells, and then they get shipped back. And then while the patient's waiting for that infusion, they receive 3 days of low-dose, gentle chemotherapy called lymphodepleting chemotherapy with fludarabine and Cytoxan to make space for the T cells, and then a couple days of rest to let that chemo filter out of their system, and then they get their T cell infusion. Now, the two main side effects that we watch for, especially the first couple weeks, and often this is done in the hospital. Some programs do these outpatient, but they have to come in every day and be monitored very closely because of these risk of side effects. One would be cytokine release syndrome, or CRS. That's basically when these T cells go in and blow up the targets, and they release chemicals called cytokines, like interleukin-6. And that's the same thing that happens when you get the flu. So like the fevers and chills and body aches, that's basically what CRS is. But in severe cases, it can lead to low blood pressure, low oxygen, or organ dysfunction if it's not brought under control. And then neurotoxicity, we typically call this ICANS, or immune effector cell associated neurologic syndrome. Basically, these cytokines are causing brain swelling, and they can cause anything from confusion to tremor to even in worst case seizures. And because of that risk of a seizure, which is actually much more common in lymphoma and leukemia CAR T, we don't typically see much of that in the myeloma, but just because of that risk, the patients aren't allowed to drive for 8 weeks after the CAR T. We actually have data and surveys that we're going to present at ASH this year that proposes moving that to 4 weeks instead of 8 weeks, but right now it's 8 weeks, and they have to kind of stay within at least within a couple hours of the medical center for the first month because of the risk of these side effects. This is an example of a patient's handwriting on day 4 that was normal. By day 5, their handwriting turned to gibberish because of some CRS and neurotoxicity. They got a dose of tocilizumab, which is an anti-interleukin-6 antibody, and that resolved within a few hours, and by the next day it was normal again. So most of these symptoms are kind of transient. They're resolved within a day or two. Now, certainly a few % of patients can have more severe or long-lasting. There are 6 different CAR T cell products that are already FDA approved and on the market. 4 of these recognize CD19, and they're used for B-cell malignancies like lymphoma or acute lymphoblastic leukemia. So we had to come up with a different target for myeloma, which is BCMA, or B-cell maturation antigen, and that one is the target for both ide-cel and cilta-cel. And here's the reason. So CD19 is expressed on all of these earlier B lineage cells, like Dr. Afro mentioned, or actually Dr. Khan mentioned. The plasma cells are derived from B cells, but they're kind of terminally differentiated, and so they have different antigens compared to the earlier B cells. And so BCMA is kind of a plasma cell, pretty specific marker. So yeah, are we using these CARs to turn the corner now? We have 2 different ones approved, as I mentioned. First, for late line relapse after 4 or more prior lines of therapy. Just this year, they've now been approved for early relapse. So with cilta-cel approved after 1 prior line of therapy, and ide-cel approved after 2 prior lines of therapy. And so I'll just go through a couple slides on what led to their first approval. So this was the KarMMa study. That was the registration study that led to the FDA approval of ide-cel in relapsed refractory multiple myeloma. This study required 3 or more prior lines of therapy, although the patients on the study actually had received a median of 6 prior lines of therapy, so very heavily relapsed patients. In this population, we've already done studies before this showing that the expected outcome with any other standard therapy before CAR T would have been maybe 25 to 30% chance of getting a remission, and then once they get a remission, maybe 3 or 4 months before they need to move to something else. So very difficult to treat population. This data we published in the New England Journal in 2021, and that, as I mentioned, led to FDA approval because we saw unprecedented responses. 81% of patients that received the target dose of 450 million achieved a remission. 39% achieved a complete remission. We do see the side effects that I had already mentioned, like cytokine release syndrome in the majority of patients, but it was mostly low grade. Only 5% ended up having to have low blood pressure and low oxygen and things like that. So a minority of patients, and again, these are patients with widespread full-blown myeloma relapse that had tons of myeloma, so much different than when we are talking about early relapse. But fortunately, even though studies of CAR-T and lymphoma show a lot of patients with neurotoxicity, only 18% of patients had any neurotoxicity at all, and mostly was low grade. Only 4% had higher grade neurotoxicity. And the overall survival, which would have been expected to be less than a year for any other treatment in this group, was 24.8 months. And the most notable thing is if patients were lucky enough to achieve a complete response with ide-cel, they had a 41% chance of still being in remission at 2 years. And we just a few weeks ago presented a 5-year update showing that about 17% of those that achieved a CR are still in remission at 5 years, which equates to about 6 to 7% overall. So most of these patients do relapse within 1 or 2 years with ide-cel. So it's not a complete home run that we were hoping for, but at least some of these patients can have very meaningful benefit, like the patient I mentioned earlier. And what happens with ide-cel in the real world? So we have all these patients that are fit enough for clinical trials, but what about regular patients that can't qualify for trials? So we're part of the U.S. Myeloma Immunotherapy Consortium, which at the time of this publication last year had 11 different institutions across the country. Now there's about 20 that are sharing data from all of the outcomes from CAR T-cell therapy so that instead of just a bunch of little tiny studies, we can have one big study for all of these different questions that we're asking. And we said what happens in the real world with ide-cel after FDA approval. And out of the 159 patients evaluated, we noted that 75% of those patients would not have been eligible for clinical trials because they were too sick, had too many other medical conditions we call comorbidities. But fortunately, the outcomes were almost identical to the clinical trial, with 82% experiencing low-grade cytokine release syndrome. Only 3% had higher grade. Only 18% had neurotoxicity of any grade, and only 6% had higher grade neurotoxicity. The response rate was 84%, and 42% achieved a complete response. So very similar to the trial. But again, most of those relapsed within 9 months, 9 to 12 months, essentially. But we did see that it was safe for patients who have kidney failure or renal impairment. We did also note that prior BCMA-directed therapy impairs the outcomes after CAR-T. And we also noted that patients that are older can safely receive, especially ide-cel CAR-T. This was our real-world study looking at prior BCMA-directed therapy. So some patients are like, okay, should I get a bispecific? Should I get a CAR? Which one should I do first? So this was some of our first data trying to help determine which order we should go in. So this was the patients who received ide-cel in the real world who had had prior BCMA-directed therapies. In this study, we saw patients, there were only 5 patients who had received prior BCMA CAR-T, and all 100% of those patients still responded to ide-cel. So prior CAR-T did not have any impact on the responses, but prior bispecifics only didn't really affect the response rate. Those 7 patients, out of those 7 patients, 86% still responded. However, the most notable thing is those remissions did not last very long. So if you look at the green line here, so only the remissions only lasted about 2.8 months for those that had prior bispecifics against BCMA. So prior BCMA bispecifics before BCMA CAR is not a good idea in general whenever possible. But notice the ones that had prior CAR-T had no impact on their progression-free survival, their duration of response. So that is one take-home message. And I think Dr. Afro has already gone through this data. I just wanted to point out that those that had prior CAR T still could respond very nicely to bispecifics like teclistamab. But although the duration of response may be slightly impaired, but not as nearly as bad as when you do bispecifics before CAR T. So if ide-cel works well, you know, in the late line relapse, what about earlier line? So the KarMMa-3 study looked at comparing actually patients who had received 2 to 4 prior lines of therapy, and then they randomized these patients to either ide-cel CAR T or standard of care. And the standard of care options were, the investigator could choose between 5 different chemotherapy combinations: Dara-Pom-Dex, Dara-Velcade-Dex, Ixazomib-Revlimid-Dex, Kyprolis-Dex, or Elo-Pom-Dex. So they could choose based on which ones the patients had received before. And then the patients that did not receive CAR T were able to cross over if they relapsed and get CAR T later. But the patients that received ide-cel CAR T had significantly higher chances of achieving a response or remission, overall response rate of 71% compared to 42%, and the remissions or progression-free survival was also significantly longer with CAR T. Although, notice it's not necessarily a home run still. We have just a little tail on that curve on the right there that shows that a small percentage of patients are still in remission at 3 years, but the majority with ide-cel do still relapse within a couple of years. But based on this significant improvement compared to standard of care at the time, ide-cel was FDA approved this year to give to patients who've had 2 prior lines of therapy that are triple class exposed, so a PI, IMiD, and a CD38. So ide-cel is one tool we have in our toolbox. What about other options? So we have CARTITUDE-1, that was the first study that led to approval of cilta-cel, or Carvykti, for relapsed myeloma. So the CARTITUDE-1 study was designed much like the KarMMa study. It had patients who had received at least 3 prior lines of therapy, but the actual study patients had received a median of 6 prior lines of therapy. So again, very refractory, heavily relapsed patient population. And one other difference with cilta-cel is it has 2 BCMA binding domains instead of 1. We don't know exactly what makes the difference here, but that is one difference. And the CARTITUDE-1 study showed 97.9% of patients, again, with a median of 6 prior lines of therapy, achieved a response, and over 82% had achieved a complete response with no detectable monoclonal protein in the blood or urine, and 95% achieved over 90% reduction of their M protein. This translated into a median progression-free survival of 34.9 months. So at 3 years out, roughly half the patients were still in remission, and there's still many ongoing remissions from that. The safety was a little bit different. We did see the majority of patients with low-grade cytokine release syndrome. High-grade was, again, pretty rare. Neurotoxicity, pretty similar at about 20%. But one difference in this study with cilta-cel was that there were 5 patients out of these 97 or so that had a movement disorder like Parkinson's or Parkinsonism. And so that was some cause for concern. And then also 7 patients developed some other, what we call delayed neurotoxicities, like cranial nerve toxicity, like a Bell's palsy, essentially. And so one of the things that Janssen, the company that led this trial, did next was look at the factors that were associated with these delayed neurotoxicities and movement disorder, and decided that the risk factors were patients at a high tumor burden, so a lot of myeloma on board, which also correlated with high-grade cytokine release syndrome and high-grade, early on neurotoxicity. So it was decided to try to better cytoreduce these patients to try to prevent that. That means getting them better bridging therapy between the collection and the infusion. We want to try to get their disease down as much as possible to try to prevent that. And then if they get a fever, instead of just giving Tylenol, we would actually treat it more aggressively and try to make sure they don't have a higher grade cytokine release syndrome. So the next study that led to the early line approval of cilta-cel is called the CARTITUDE-4 study. This was for patients who had received only 1 to 3 prior lines of therapy, so early relapse myeloma. They were refractory to lenalidomide, so most of these patients had had often a stem cell transplant and were on lenalidomide maintenance and then relapsed. Most of these had not received a CD38 antibody like daratumumab. But it randomized patients to either cilta-cel CAR-T or investigators' choice of standard of care, and they could choose between Pom-Velcade-Dex or Dara-Pom-Dex, which are pretty common regimens. And the complete responses were over 73% in the CAR-T group compared to 21.8% in the standard of care. And with the recent follow-up at the International Myeloma Society meeting a few weeks ago, we saw at 30 months, 59.4% of these patients were still in remission and alive, compared to only 25.7% that received the non-CAR-T therapy. And also, importantly, now we're seeing an improved overall survival with early CARVYKTI. So at 30 months, 76.4 were still alive compared to 63.8 that received non-CAR-T therapy for their early relapse. Now, we're always taught you were never supposed to compare two groups from two different studies, so that's what I'm doing here. But basically, I just wanted to give you an example of the outcomes from the late line CARVYKTI versus the early line, showing that with CARTITUDE-4, with the earlier relapses, we are seeing better survival and longer remissions compared to if we save it until after 6 prior lines of therapy. Let's just put it that way. And just to highlight, we don't have a direct clinical trial comparing the two CAR-T products, ide-cel versus cilta-cel, and KarMMa-3 and CARTITUDE-4 were similarly, were not exactly similar. The KarMMa-3 study was, again, for patients who had 2 to 4, so not the 1 to 3 that we saw with CARTITUDE-4. And the majority of these patients in the KarMMa-3 that got ide-cel were heavily refractory and what we call triple class refractory, whereas only a minority in the CARTITUDE-4 were triple class refractory. The majority of the patients in KarMMa-3, or 95%, had received a CD38 antibody like daratumumab, whereas only 24% had received a monoclonal antibody like daratumumab in the CARTITUDE-4. So it's different patient populations. Also, we don't really see the delayed neurotoxicity with ide-cel, but after the mitigation strategies of better, earlier treatment and better, reduction of cytokine release syndrome, et cetera, fortunately, the risk of Parkinson's symptoms was significantly reduced. Only 1 patient out of these 208 developed some symptoms of Parkinsonism, so they calculated at about 0.6% of the patients that were evaluable. So not 0, but way better at this point. They did still see about 8% risk of cranial nerve palsy, so like a Bell's palsy or facial droop. That is typically treated with steroids and resolves within a month or 2, but obviously can be quite scary if that happens. So really a very minority of patients would have a long-term neurologic issue, 0.6% basically. So something to, some patients use that to make a decision, but at least significantly improved when we used that earlier. So what about, how do you decide, between CAR-T, bispecifics? We already gave some data on that. Just, sometimes it's logistics and, patient preference, but, CAR-Ts are personalized, bispecifics are off the shelf. You don't have to wait for production. They both target the same things typically. Potentially the CAR-T cells can live in the patient and sort of act as like a living drug, whereas the bispecifics, you have to keep giving them. They get, weekly or every other week shots long-term as long as they're working. CAR-T cells are dependent on T cell health at the time that you collect them, and so it doesn't matter what you give for bridging right after that. You could potentially give something stronger after the collection, whereas bispecifics depend on the health of the T cells constantly because you're continually training different T cells to recognize the myeloma. The CAR-T cells require no driving for 8 weeks, whereas bispecifics, well, some of them you're only in the hospital for 3 days and can start driving as soon as you're out of the hospital. So it doesn't require quite as much caregiver support. So some of the things that we use to decide, among other things, like how is the disease biology? Are they having a rapid relapse? Are they having just a very slow, gradual relapse? Is their disease exploding and having plasmacytomas everywhere, or is it just the light chain elevation? You know, do they have a family? Do they have a caregiver support? Do they have anyone that can drive them at all? Because if they don't have anyone that can drive them, it's going to be very difficult to get a CAR T cell therapy. You know, how far do they live from the center that does CAR T? Do they need to stay locally? All of these things can add up. You know, so now, you know, we're blessed to have these options. We could potentially give CARVYKTI, CAR T cells to patients who had their first relapse now. But does everyone need this? I don't think everyone needs them to be given in second line. So for example, these, you know, definitely patients that are younger, fitter, don't have any neurologic issues, especially those that have early relapse. You know, some would say less than 18 months is a no-brainer, but certainly within 2 to 3 years, definitely going to be stronger case for going ahead and being more aggressive with CAR T cell earlier. Those that have, you know, these extramedullary plasmacytomas and rapidly growing disease, those definitely would be great candidates. But if someone, for example, you know, was older, and let's say even if they had relapse disease, but they were more frail, you know, ide-cel is definitely a great option for these patients, even if they have a lot of, you know, kidney failure or other diseases. And you know, we recently gave this to a 90-year-old and they did great. So it's, you know, very well tolerated. But let's say we have a patient who's been in remission, you know, they had VRD and an auto transplant and they've been in remission 5 or 6 or more years, you know, and they've never had a CD38 antibody. Do we really need to jump in and give them a CAR T? Probably not, you know, unless they really want to be aggressive. It is an option to discuss, but, you know, they could probably get, you know, daratumumab and POM or some other sort of CD38 combination that might last for many years for those patients. So you don't have to be drastic and jump into this for everyone. It's an individualized discussion, you know, between the patient, the physician, the caregiver that's also providing a lot of the support. So that is definitely a big factor. So some of the summaries here. So we have 2 different CAR T cell products that are approved for relapse refractory myeloma, both ide-cel and cilta-cel. Both were initially approved after 4 prior lines, but now for earlier line, the real world data shows that older patients and patients with a lot of comorbidities can still benefit from these therapies. Cilta-cel is now approved after 1 prior line of therapy, which means giving it in second line. It does require prior proteasome inhibitor and to be, quote, lenalidomide refractory. Abecma or ide-cel is approved after 2 prior lines of therapy, including those that have had a proteasome inhibitor, IMiD, and a CD38. The KarMMa study population was highly different from the CARTITUDE-4, so it's not really the same populations, but you know, for the most part, whenever patients have a more aggressive disease, you know, it does look like cilta-cel has the longest remissions that we've seen in this space. We do also, you know, we initially worried about the delayed neurotoxicities, but now that we have better strategies for getting the disease under control between collection and infusion and a more aggressive treatment of the cytokine release syndrome, we are seeing a lot less risk of neurotoxicity. And we do want to point out that prior BCMA bispecifics does significantly impact your outcome after BCMA CAR T. At least the current ones. Now, if there's a different target, you know, those it's unknown, but at least the current ones. And so one of the things that we're doing now, you know, we know that CAR T works well late and early, but what about the earliest, using it in frontline? So this is one of the studies that we've just opened at UT Southwestern called the CARTITUDE-6 study. It's open, you know, around the world, but we just opened at our site as well. So this study is looking at newly diagnosed patients who have not been treated yet. And all of these patients, well, these are patients that are transplant eligible, so usually younger, fitter. They're randomized to either CAR T or autologous stem cell transplant. And both of the groups receive quadruplet induction therapy with Dara, Velcade, Revlimid, and Dex for 6 cycles, and then half the patients get Carvykti CAR-T. The other half get autologous stem cell transplant, and then 2 years of lenalidomide maintenance in both groups. So it's going to be very interesting to see which one comes out ahead. I'm a transplanter, but I'd be happy to give it up if CAR-T works well. So these are just some of the studies that we have ongoing right now, looking at different CAR-T cell products. So we have CAR-Ts against GPRC5D, just like the target for talquetamab. The phase 1 that we participated in was just presented at International Myeloma Society meeting with 96% response rates in refractory disease. Phase 1 just finished, and we're just opening the phase 2 that will be geared toward trying to get it FDA approved later. We also have the Arcelex or ddBCMA, now called anito-cel. That one, the phase 1 just closed to enrollment. Phase 2 just closed to enrollment. We're about to open phase 3. But the phase 1, had 100% response rates and no patients with Parkinson's symptoms. So very promising early data. And I mentioned the CARTITUDE-6 already. We also have CARTITUDE-2 cohort G for patients who are not quite where we think we want to push them toward a stem cell transplant. We can potentially get them CAR-T frontline without randomization. In this group, they get daralindex for 4 cycles, followed by Carvykti CAR-T in frontline. So exciting times to see if we can get even better response. And one of the rationales is that if we collect in both of these last 2 studies, that if we collect their T cells before they start treatment, can we get a more fit, more functional T cell that can lead to more longer lasting CAR T cell product. Thank you.